Now we're finally ready to build new DNA. And what we use to build new DNA are DNA polymerases. They polymerize DNA. They put nucleotides together. They catalyze the synthesis of DNA. Yay, we're going to make DNA by adding nucleotides to a growing chain of nucleotides. See what I mean? They can't grow from scratch. They have to add nucleotides to a growing chain. The growing chain, folks, is this little primer that's already laid down. Let's take a look at a picture of DNA polymerase in action. And then we have a couple of things that are starred here. This means they must be really important, all right? Turns out, by the way, there are different kinds of DNA polymerases down here. For our purposes, let's just say DNA polymerase, a type of DNA polymerase, right? And DNA polymerase builds new DNA pretty quickly, much more quickly in bacteria, but still it can lay down 50 nucleotides a second in eukaryotes. So here's a picture of, let's orient you a minute, a little piece of DNA. And in this little piece of DNA, we're adding a new nucleotide because we're building DNA in this picture. What we have, first of all, is a template strand. That's the original strand that we've unwound, and we're using it as a template to build a new strand of DNA. Also notice that these are the template strand and the new strand run in different directions. They're anti-parallel to each other. The bases pair up so that A pairs with T and C pairs with G. And it looks like we need to add a base here. We're trying to add the next nucleotide, right? And we're adding it to the 3' end of a growing chain. So here's our chain. We're adding it down here at the 3' end, okay? It's the only place that DNA polymerase can add. It can't add up here. We can't put something new up here, okay? Can't do it. It can only add nucleotides at the 3' end of a growing chain. So what DNA polymerase does, and here's our enzyme here, this is like a reaction shown in the picture, right? DNA polymerase finds and inserts the appropriate nucleotide. And so what it does is if it sees an A, it goes, oh, I need to add a T nucleotide. It adds a T nucleotide to this 3' end of a growing chain. And as it adds it, it really adds, it's not quite a nucleotide, it's called a nucleoside triphosphate because it has 3 phosphates sticking out there. It's not going to keep all those phosphates, it's only going to keep 1. 2 of the phosphates are removed, as you can see, and they're kind of a source of energy. So what it does is it adds the nucleotide, it makes this phosphodiester bond, or that bond right there, happen, and now it's ready to add the next nucleotide. What would be the next nucleotide that comes in here? Well, the template strand has a C. What should I add? Well, I'm going to add a nucleotide with a G, right? And it can keep building, and it builds the new DNA 5' to 3' on the new strand. So it only can build in this direction. It cannot add things up at the top end. Okay? It can only build 5. We always say it builds 5' to 3'. Okay, so that's the second important point. The first important point is you have to have a starting point. You have to have nucleotides already in place to add to. That's the primer. You also can only build new nucleotides in a 5' to 3' direction. You can only keep adding nucleotides on the 3' end. Okay, that's why it grows in the 5' to 3' direction. That's how DNA grows. Okay. All right. And so if we were building on this molecule here, and we had, let's just put these in, T, A, B, G, C. And down here you have A, T, T, C, C, G, right? If we have this molecule here of DNA that's been unwound at its origin site, remember it's running 5' to 3' on the top strand and 3' to 5' on the bottom strand. We would first need to build an RNA primer. Okay, I'll build that in red. The RNA primer lays down in a 5' to 3' direction. So on the upper strand here, the RNA primer builds a short sequence of RNA, in the 5, this is 5' to 3' direction. It builds and it matches up with these little guys. Now DNA polymerase, let's do a different color for DNA polymerase. DNA polymerase can come along and build new nucleotides, DNA this time, that build in the 5' to 3' direction. So again, you can put in your 5' and 3', and they would add nucleotides that complement, the A, the T, the A, and the T. So we'd put in a T, and an A, and we would keep going. And as this replication fork unwinds, this DNA polymerase, let's call this guy DNA polymerase, can continuously replicate the DNA on this strand. It keeps on moving. As the unwinding happens, the DNA is replicated and more DNA is replicated, etc. Hey, DNA polymerase is building new DNA. We're making a new strand. Now, let's see if I'm here. Yeah. The DNA polymerase can replicate in the 5' to 3' direction only. So what that means is at any replication fork, one of the templates or one of the new strands of DNA that's being built is being built in a continuous fashion. We needed a primer, but once we put the primer in place, and I'm going to write down that that's the primer there. Once we put that primer in place, we could build DNA in a continuous way, 5' to 3' as we opened up our replication fork. Right? No problem. The other template strand of DNA is running in the opposite direction. And at this replication fork, as we open it up, DNA polymerase cannot simply easily build DNA continuously along this fork. It cannot continuously lay down nucleotides going this direction because it can't build DNA in the 3' to 5' direction. So let's look at some pictures, and I'll try to draw this out again. Okay, this is kind of a strange picture, but it's a picture of a little part of a replication bubble. So here's our origin of replication up here, and here's our bubble. Overall, we've got DNA being replicated in both directions from that bubble. And we're looking at what we would call the leading strand of DNA replication. So far, that's all we've looked at, this little box here, right? This little part of a replication bubble. We laid down a primer. That's what's in red. Then DNA polymerase came along, and DNA polymerase then builds new DNA, and it builds it all in the 5' to 3' direction, and it can build DNA continuously on the leading strand of DNA replication. So we see this beautiful new strand of DNA being built in the 5' to 3' direction. The problem is what's going to happen over here on the lagging strand, okay? What's going to happen on this side of the molecule of DNA? That's called the lagging strand. Let's take a look. What's happening at the lagging strand? That really is our question. What is going on at the lagging strand? Okay, let's fill these in and then we'll take a look. So in order for any DNA replication to happen, in order to start replication, we always need a primer. So a primer must be in place. We have to build a primer first. That's made up of RNA. The lagging strand is then synthesized in short pieces or fragments. These short pieces are called Okazaki fragments. Okazaki fragments. They're named after the person who discovered them, all right? So we're going to see that on the lagging strand, there's some problems. You can't build DNA continuously at the replication fork. You have to build it in little pieces. All the pieces are being built 5' to 3'. Eventually, though, you have to join together all the little pieces into one continuous molecule of DNA. All right, so we need to take a look at the lagging strand. This is what we're really looking at down here. We're looking at this replication fork. Okay, we've already studied, this part of it, the leading strand. We added a primer, and then we build new DNA in a 5' to 3' direction. Pretty easy. But over here on the lagging strand, as you're opening up your replication fork, you have to lay down a primer, and the number 1 there means that's the first little piece that's built. A primer in red is laid down. Then you have to build DNA in this direction because you can only build DNA 5' to 3'. Then you have to unwind the replication fork a little more and lay down a second primer that's in red by number 2 and build more DNA in the 5' to 3' direction. Notice that the template strand runs the opposite direction. And then you have to do it again and again and again until you get you're building DNA in little chunks. Notice also that at the other side of the replication fork, just for fun, to make things more complex, what was the leading strand now becomes the lagging strand because you have to build DNA in the 5' to 3' direction. You have to build short fragments of DNA. Over on this side of the replication bubble or the replication fork, after you've laid down a primer here, you can build continuously 5' to 3'. So at a single replication bubble, each new strand being built on one side of the fork is called the leading strand, and on the other side is called the lagging strand. Crazy, but true. Think about how this is happening inside your cells. Every time your cell has to build a new cell, it has to replicate all of its DNA, all 46 chromosomes of its DNA in this crazy manner and not make any mistakes. And so let's draw this in. So this is going to be my leading strand, and I know that based on the direction of my molecule here. But what happens is after a primer is laid down, I better lay down a red primer. After a little primer is laid down, 5' to 3', then DNA polymerase can come along and build new DNA continuously in the 5' to 3' direction. I'm going to put my polymerase there, and it builds boom, 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 boom. It's building new DNA continuously. That's the leading strand. The lagging strand is much more complicated, right? That says lagging. It kind of says lagging. So as we first open up the replication fork, DNA polymerase is going to lay down, RNA polymerase is going to lay down a primer in the 5' to 3' direction. Then DNA polymerase can come along and build new DNA, continuing on the primer and building new DNA, okay? The replication fork opens up a little more, and we have to lay down another primer, 5' to 3', okay? Then DNA polymerase can come along and build new DNA, 5' to 3'. And then we open up the replication fork a little more, and we're going to have to lay down a new primer, 5' to 3'. And then DNA polymerase can come along and build new DNA, 5' to 3'. So we get little fragments of DNA being built, maybe 100, 200 nucleotides long. That's the lagging strand. We're going to have some problems. We have to eventually have a complete molecule of DNA. We're going to have to get rid of anything that's red in our drawing. So we're going to have to get rid of all of the primers, and we're going to have to connect all of these little pieces of DNA, right? Here is a picture of the replication of the lagging strand, and it's awkward. It's an awkward drawing. That's all I can say. I think there are some animations on our Pearson site that would be worth doing. But let's just run through all of my words here next, right? So the first thing that has to happen is helicase unwinds the DNA molecule. Primer lays down a short new RNA sequence called a primase, lays this down, called a primer. It runs in the 5' to 3' direction, and then DNA polymerase, shown here in orange, makes the first Okazaki fragment, builds a little fragment here, and then the polymerase detaches, DNA polymerase detaches, and now we need primase again to make a second primer. Here's our 5' to 3' primer. And then, here it is, right here, our second one being laid down. And then DNA polymerase comes along and builds new DNA. And then it happens again and again and again as we open up the replication fork at the lagging strand. Eventually, we have to get rid of all of the primers and connect all of the DNA together. And this note here says a different DNA polymerase removes the RNA primer. Oh, so we have to get rid of it. A different DNA polymerase, shown here in yellow, removes the primer and replaces it now with DNA nucleotides. It can build from its other DNA, and it kicks out the RNA, puts in new DNA. So another type of DNA polymerase, don't worry about the type. But finally, we're left with a whole bunch of pieces of DNA, but they're not connected to each other. There are little breaks or gaps wherever there had been a primer, right? And so there is another enzyme yet called ligase, and that's shown in green here, that has to ligate or glue together the newly synthesized DNA fragments. So these are the things that we just mentioned. Primers must be removed. You got to get rid of the RNA and put new DNA in its place. After the primers are built and used, then they need to be removed. DNA polymerase removes them and replaces the primer with DNA. But it cannot join the final nucleotide to the already in place nucleotides. They can't make that final connection. And then there's an enzyme called DNA ligase that joins them together. Okay? Yes. I like this summary slide. It's sort of static, like our video has been, but it kind of helps you understand what's happening on the lagging strand in particular. It recognizes that you have to recognize that this is all happening continuously. One of the things I wanted you to notice is it names DNA polymerase as DNA polymerase 3 and DNA polymerase 1. We're not going to worry about the Roman numerals here. We're just going to call them a type of DNA polymerase, right? There's also a ton of DNA replication videos. This one is what I recommend, but there are lots of them to look at. So check them out. I think they will help you enormously. This is one of these things that you need to actually do to understand DNA replication. Let's see if we can answer all of these questions. I guess we'll do them in order, but I'm looking at a little section of a replication bubble. Also, it says look at the Mastering Biology animation here. That also might be good. And I think that we've already unwound the DNA. I'm guessing that the first blank there would be single-stranded binding proteins. The second blank would probably be the molecule that's unwinding the DNA. That would be helicase. Helicase does the job of unwinding the double-stranded DNA. It looks like this number 3 is DNA polymerase because it's building a strand of DNA. So this would be DNA polymerase. I'm just going to write DNA pow. Number 4. It looks like number 4 is an enzyme building a primer. So that enzyme is primase. Number 5 looks like we're talking about the newly synthesized DNA that is being synthesized in a continuous way. So that would be the leading strand of DNA. Number 6, it looks like that's a chunk of newly synthesized DNA, so I would call that the lagging strand, or it could be called an Okazaki fragment. Oh, maybe we should write Okazaki fragment, probably. I'm not going to rewrite that. Number 7, it looks like we're dealing with another DNA polymerase here. That's building our DNA. And number 8, we have an enzyme that's ligating or gluing together the newly synthesized DNA fragments. That has to be DNA ligase. Look how much it takes to build new DNA. Look at all the molecules involved that we've been talking about. Building new DNA is complicated. That's the end of video 2, and in video 3 we'll continue with this and look at just a couple other topics that are found in chapter 16. That's tough. If I were you, I recommend definitely looking at animations of DNA replication, trying to draw it out, because that's going to help you enormously in understanding it. I think that's it for now. I'll talk to you later.